Hello everyone, I'm Rami-Petteri Apuli, a final year PhD student for Professor Per Ingvarsson at Department of Plant Biology at the Swedish University of Agricultural Science. I am here to talk to you a little bit about my work with deleterious load in Populus tremula across an active admixture zone in Sweden. I will not be going into deleterious load very thoroughly here. However, I will be giving you 3 points that will hopefully help you understand my work here. First of all, deleterious load has been often defined as all segregating and fixed alleles that lower fitness of a population. However, this is kind of difficult to measure, the fitness I mean, and it's even more difficult to measure the effect of an allele on it. So deleteriousness is usually inferred from amino acid changing mutations from multiple species alignments. And that is how we have done it too, using this bad mutations pipeline. Then we heuristically defined the deleterious allele in the deleterious loci. Deleterious load has been also found to increase with geographic distance to the center of origin, whether that be the population origin or the species origin. And this is caused by the successive bottlenecks, founder effect and drift that accumulates during the range expansion. And deleterious load has also been found to increase with selective sweeps in both the selective sweep window and the immediate surroundings. Most deleterious alleles are also recessive, which means that they need to be in homozygous form to have a negative effect on an individual. So, what makes the Swedish Populus tremula so interesting to study is the fact that the south and north subpopulations have different refugial origin. Now, I will be calling these subpopulations populations for clarity's sake in the coming slides. So, the north Swedish populations, they originated in an eastern refugia, while the southern populations originated from a central European refugia. And now these refugial origins are admixing in the central populations in Sweden. And this is kind of supported by this structure plot I have here. If we look at the north population, we can see that there is quite a large proportion of Russian influence in the north population, whereas the Russian influence is far lesser in the south and central populations. However, opposedly, there is a lot of Latvian influence in the south and central population, while that is far less in the north population. So following this, I will be using the Russian and Latvian populations as proxies for the Eastern and Central European refugia respectively. And the final thing that I will be looking at is this FT2 northern allele, which introgressed from Russia to this north population and was swept to fixation in it. So this sweep is a very strong, well-defined and well-studied sweep in chromosome 10 at around 4.4 to 5.1 megabase pairs. Okay, going into the results here, I just want to point out that I am only using deleterious alleles with frequency higher than 5%, in these results. This is quite simply because it is going to be easier to see the differences in the admixing populations using the higher frequency alleles. Okay, so on the left here I have plotted the deleterious homozygote count divided by population deleterious locus count for each of the individuals in each of our populations. For going onwards, I'm going to call this simply deleterious load. And in this plot, the interesting part is here, between the South population and the Russian population. As we can see, the central population has significantly lower deleterious load than the South population. And similarly, the North population has significantly lower deleterious load than the Russian population. However, the Central and the North population are not significantly different. So what this tells us is that there is evidence of purging of deleterious load in this admixture zone. However, what is somewhat unexpected is the fact that there is purging also happening in the North population. So this admixture zone and this purging seems to be happening far more north than what we initially expected. Okay, let's look at the FT2 sweep next. We can already see that the pattern is not the same as in the whole genome. We can still see that South has more deleterious load than central population, and that difference is significant. However, in the case of FT2 sweep, the North population also has more deleterious load than the central population. And what is also interesting is that there is no significant difference in deleterious load between the Russian and the North population. So there might be some purging happening in this window, but now the purging is happening only in the central population. So there is no purging happening in the deleterious load in the North population. So this sweep has resisted purging. So this is most likely because the FT2 northern allele is likely important for adaptation to northern conditions. So, just to summarize, there seems to be release of frequent deleterious homozygosity and purge of deleterious alleles in both central and north populations, which is kind of to be expected from the relationships and demography of the populations. However, we do not see similar purging in the FT2 sweep, which is likely due to the importance of the FT2 northern allele to adaptation to northern conditions. Here is the references I used for this presentation, where you can learn more about deleterious load and how it behaves, as well as the FT2 sweep. And I finally want to thank you for your attention, and I want to extend a huge thanks to all Ingvarsson group members and ex-members for their input and help in putting together this presentation. If you have any questions, comments and suggestions, feel free to ask them in the Q&A, and if you need to get in contact with me, you can see my email address down below.
Hi, I'm Mitra. I'm a postdoc at UC Davis, and today I will be sharing with you some of my recent work looking into the architecture of adaptive evolution under novel selective pressures. Decades of common garden and provenance trials in trees have provided remarkable evidence for local adaptation at the organismal and the physiological trait level. Recent studies making use of gene expression traits have provided similar signals of local adaptation. But contrary to the organismal level traits, transcriptome profiling have the potential to evaluate several hundreds of traits simultaneously and so can provide a more holistic picture of the architecture underlying adaptive evolution as well. So today I want to share with you the utility of combining transcriptomic profiling with a common garden design to evaluate not only signals of local adaptation and of adaptive plasticity, but also to make use of these co-expression patterns to ask questions about the architecture of adaptive evolution under different environmental conditions. To do so, I made use of these range margin populations of Pinus strobiformis or southwestern white pine. And as you can see from the figures, from the green range map, as well as from the picture on the right-hand side, these populations occur on extremely fragmented sky island ecosystems that are separated from each other by this vast arid desert ecosystem not hospitable for these high elevation conifer species. Additionally, climate change predictions indicate that these regions will experience warmer and drier conditions, as well as increased frequency of aseasonal frost events. So altogether, it makes these populations an interesting system to ask questions about signals of adaptive evolution, but also to ask whether these populations have the potential to adapt to novel selective pressures that will be imposed by changing climatic conditions. So as I had mentioned earlier, I approached this by combining transcriptomic profiling and a common garden design. I made use of two common gardens which were located in the north rim of the Grand Canyon. White Pocket is a low elevation, drier site. Bear Springs is a high elevation, colder site that receives most of its precipitation in the form of snow. Within each of these gardens, I sampled needle transcriptome from 90 seedlings that represented a mixed sieve family design and had maternal trees that originated from 10 populations that in the map on the right-hand side is represented by these cyan circles. Using these needle transcriptomes, I generated a de novo transcriptome assembly, and after a series of filtering steps, ended up with 27,000 transcripts, which for the purpose of today's talk, I will be equating to 27,000 quantitative traits. For each of these, I fitted a mixed effect linear model, which took into consideration aspects of our design, as well as aspects which were specific to the RNA-seq protocol. Using the variance components obtained from this model for each trait, I estimated the degree of trait divergence, which is QST, and ranges from 0 to 1, with a value close to 1 indicating that the populations experience strong divergent selection. But a value of 1 could also mean that the populations have very restricted gene flow amongst them. So in order to tease these two apart, I generated an independent data set by making use of dd-RADseq and genotyped each of the same set of 10 populations at SNP markers and estimated genome-wide levels of neutral divergence or FST. I then compared QST to FST using a statistical framework to ask questions about adaptive evolution at each of the gardens and also of adaptive plasticity. With this, I ended up with 3 EQST categories. This is further highlighted in this schematic, and these will be the focus of my talk moving forward. Y-axis here is representing the degree of among population differentiation, and FST here is represented by the black ovals with the confidence interval. QST for each of the categories and the transcripts falling under them are represented by differently colored box plots. So overall, across the two gardens, by using this QST-FST comparison, I identified 334 transcripts which were conditionally adaptive in White Pocket, represented by the orange, 234 that were conditionally adaptive in Bear Springs, and about 26 transcripts that exhibited signals consistent with adaptive plasticity. So with these EQST categories, I can move on to evaluating differences or similarities in the architecture underlying adaptive evolution. I did this by making use of co-expression networks. Here, the circles are representing the transcripts, and the connections between them represents the degree of covariance in their expression patterns. Groups of transcripts that strongly covary in their expression are classified into a module, which here is represented by the blue, red, and the green modules. We combined this co-expression network evaluation and the topological properties of it, along with the EQST categories that I had identified in the previous slide, to ask questions about the genetic architecture of adaptive evolution. Specifically, we expect that the EQST transcripts should have, in general, weaker connectivity or concentrated away from the core of the co-expression network. Here I'm equating connectivity to level of pleiotropy as estimated from the co-expression network. And in general, we expect pleiotropy to be costly because genes or transcripts which have high levels of connectivity can modulate the entire co-expression network. And such large-scale modulation would probably be costly and deleterious in nature, and hence we do not expect it to exhibit strong among population variation. However, under certain conditions, when the populations are experiencing or are exposed to novel selective pressures, we expect that the EQST transcripts may exhibit strong connectivity or here pleiotropy may actually be favored despite the cost that it imposes in the long run. So in order to evaluate this, I made use of co-expression modules that were identified at each of the two gardens and then asked whether the EQST transcripts were over or underrepresented in the core of the co-expression network and that of the module. And here identification of core is basically based upon the level of pleiotropy for a given transcript identified through a measure of connectivity with everything else in the network. For each garden, I only made use of the conditionally adaptive set of transcripts as the EQST category. Okay, so in this figure, the box plot is representing 10,000 permuted values of the relative representation of EQST transcripts in the core. And what we notice is that across both gardens represented here on the x-axis, our EQST transcripts actually tend to be over-enriched in the core of the network and that of the module. And what this represents is similar to this figure that I'm showing on the right-hand side, wherein our EQST transcripts shown by the blue colored dots are largely concentrated towards the center of the co-expression network and hence have high connectivity, indicating that they can modulate the entire co-expression network. So what we see here is pleiotropy is actually favorable. And while this seems to go against a lot of the population genetic theory, as well as accumulating studies using co-expression networks, it may make sense for our situation. Because as is shown in this PC plot, the two gardens where these trees were sampled from experienced or exposed the trees to environmental conditions that were beyond the range of anything that these populations naturally experience on average in their native environments. And so here they are exposed to novel selective pressures, which actually is quite similar to what we might expect under changing climatic conditions. So overall, here I want to conclude by stating that gene expression can be a very useful way to evaluate signatures of adaptive evolution in these long-lived species. And in trees, despite their long generation time, there may be a lot of potential for adaptive evolution to novel selective regimes, and this might happen through a shift in the architecture of adaptive evolution, giving a predominant emphasis to pleiotropic genes, at least during the initial stages of the adaptive walk. And with that, I have several people and funding sources to acknowledge. And if you have any questions, please feel free to reach out to me either via Twitter or by sending me an email. And that, thank you.
Good morning, everyone. My name is Tal Shalev. I'm going to be talking about Western red cedar and its genome and how exceptionally low genetic diversity seems to be driving strong balancing selection in a self-compatible conifer. So Western red cedar, for those who don't know, is a conifer in the Cupressaceae family. It is an economically culturally and ecologically very important species here in British Columbia. Western red cedar is the provincial tree of British Columbia and has been used extensively by coastal First Nations in their art and totems, as we can see here from these pictures out at UBC. Here we have the Truth and Reconciliation totem pole and a lot of art and sculptures at the Museum of Anthropology. And it has very unique biology, so it has a unique ecological role where it's considered a stress tolerator, so it does very well in a wide variety of environments, from very dry to very wet, warm to cold, and thus it's also a projected climate change species in Canada. As climates get drier and hotter, we are expecting to see it extend its range throughout BC. And it also has a very unique mating system for a conifer. It actually has very low outcrossing rates, and which means that it readily selfs in nature. So it has very little inbreeding depression and quite short generation times, which makes it a really interesting candidate for a lot of studies of this kind of mating behavior in conifers. All western red cedar in North America are thought to have originated from a single glacial refugium just north of California and then expanded north and inwards since the last glaciation about 10,000 years ago, and now currently range from California all the way up to the southern tip of Alaska on the coast, and have also expanded to the interior, although they don't grow very well in the mountains. And probably a good portion of this range expansion has been driven by its unique mating system, where it's been able to essentially create lots of founder populations by self-fertilizing, and thus we see that most western red cedar today have very high rates of self-fertilizing in nature, and even in, and of course in seed orchards, so anywhere from 0 to 100% in any given population. And this is actually being utilized by the ministry here in BC, where they've been able to self-fertilize these trees to up to 5 generations of complete selfing over the course of about 10 years. Now, I can't go any further without mentioning Dr. John Russell, without whom any of this would have been possible. So he was the lead breeder of western red cedar, tree improvement specialist of western red cedar in the BC Ministry of Forest, Land, and Natural Resource Operations and Development. And he unfortunately passed away 2 years ago, but so this work is dedicated to him. Now, we had some questions that we wanted to ask about western red cedar as we were sequencing the genome. And a big part of it has to do with genetic diversity in western red cedar, which is being expected to be quite low due to the fact that it has expanded from a very small population since the last glaciation. And of course, we also wanted to look at the effects of extreme selfing, since we had these really interesting selfing populations. And so we wanted to see how the western red cedar genome compares to the genome of other conifers, whether there's any evidence of genetic differentiation in a population of western red cedar, and what are the effects of extreme selfing. So trying to look a bit deeper into what genes might be under selection, and whether there's evidence of purifying or balancing selection during selfing. We used a combined short reads and linked read sequencing approach to sequence the just under 10 gigabase pair western red cedar genome. Cupressaceae genomes are typically smaller than Pinaceae genomes. This is consistent with the other Cupressaceae genome that has been sequenced so far, giant sequoia, which actually has a smaller genome than western red cedar. This is one of the most contiguous conifer genomes with an N50 of about 2.3 megabase pairs, and all this in just under 68,000 scaffolds. So this is a much less fragmented genome than some of the other conifer genomes that have been assembled so far. We assessed the completeness of the assembly and the annotation of our genome, which was generated using iso-seq full-length cDNAs, and found that the completeness for both using single copy universal orthologs to assess was very high, and in fact one of the highest of any other conifer that's been assessed today. So we have a very complete genome here. Using the genome, we were able to use a target capture methodology approach to genotype a large population of 112 individuals from across the range of the species for a number of SNP loci. And we wanted to look at how diverse really is this population of Western red cedar. And so first we looked at the genetic differentiation, so we can see how differentiated they are across all these populations. And we can see that there is a bit of differentiation of the latitudinal cline, so you can see on the left part of the axis from south all the way up to north. However, the variation that is explained by each principal component on these axes is very, very low. And so we have to conclude that there really is no significant genetic differentiation between these populations. And this can be further verified by a number of other tests that we did, such as looking at FST between the populations, which was extremely low. Effective population size, which was also extremely low, so 188 individuals for this entire range. And a pi, which is the nucleotide diversity at all loci, or all genic loci, was also extremely low, so a number of orders of magnitude lower than other conifers. And the synonymous to non-synonymous to synonymous nucleotide diversity ratio, which was extremely high, and that's indicative of low levels of purifying selection. So most SNP loci are likely being maintained by balancing selection in our population. We were able to sequence 28 full selfing lines all the way from full SIB to S5 with the same SNP approach, and we found that heterozygosity declines much faster than we expected during selfing. And further, we found that F, which is the inbreeding coefficient of the individuals in these lines, increased slower than expected. So the red dot in the plot on the right indicates where we would have expected the inbreeding coefficient to be. And this indicates to us that isolation by descent does not really fully explain the reduction in heterozygosity that we see. Rather, the observed loss of heterozygosity may be in part explained by genetic drift, suggesting that most strongly deleterious alleles may have already been purged from the genome, which is what we kind of expected based on the range expansion and the low genetic diversity that we see in the species. This abundance of heterozygous SNPs, which we see in these black dots on the left plot, could be evidence of associative overdominance of these loci. To better understand what was going on here, we looked at selection during selfing by examining all the SNPs that were deviating from our expectation of genetic drift. So they were either fixing when we expected them not to, or they were remaining heterozygous when we expected them to fix after multiple generations of selfing. And we found that most SNPs deviating from expectations of drift, unsurprisingly, remained heterozygous in most lines when they were expected to fix. So most SNP loci that we see are not under purifying selection, or rather they are being maintained by balancing selection. So there is some selective advantage to keeping a lot of these SNPs heterozygous. And this is consistent also with the large proportion of SNPs that are putatively causing missense mutations in our data set, as you can see in the left figure. So no SNPs fixed in all of the lines, which suggests that majorly deleterious alleles have already been purged. There isn't any strong selection for alleles to be fixing. And interestingly, self-pollen recognition, which is a gene ontology category for rejection of self-pollen, was significantly overrepresented in outlier SNPs, so these SNPs that are under selection. So there may be some selection to maintain selfing behavior as they're remaining heterozygous. That's something that we could pursue further. With that, I would like to thank all the people who have been involved in this project from the beginning, and the Bowman Lab Group at UBC. Thank you all for listening today.